Hi everyone, for those of you who don't know me, I've been at the Institute now for about 18 months, but I've been doing flow cytometry for over 10 years. So about 2 years ago, I really started digging into FlowJo to kind of see what more we could get out of the platform. And so what I want to run through with you today is a bit of kind of a how-to guide for putting in some metadata and doing some clustering analyses. This is particularly useful when you've got a bunch of data that you've recorded using the same panel, and you want to start figuring out what might be some differences between different cohorts, whether that be mouse or human. So I'm just going to share the screen and walk you through it. I'll turn off my video because you'll be looking at the side of my head otherwise. But if there's any, if anyone's got a comment as we go through, put it into the chat, raise your hand, or if it's really pressing, feel free to unmute yourself and just ask the question, and we'll go through it. Okay, so first of all, in terms of sorting out your FlowJo plugins, you do all that through your preferences pane here. So you've got the workflow is all available online, but you want to go down into your diagnostics pane here. Make sure that you've got the path set here for your plugins, and you've put your R path in here. So all of the details on how to install FlowJo plugins are available on the FlowJo website exchange here. So the ones that I'm going to take us through today are downsample, phenograph. I also like the FlowSOM here, which are the two clustering tools. ClusterExplorer, which helps you figure out exactly what phenotype these clusters have. And there's a whole bunch of other ones on here that may or may not be useful for what you're interested in doing. So I encourage you to sign up for updates here. There's always new ones coming on board onto Exchange, and download the instructions and go through your workflow there. So the first step here, when you've got your data that you wanted to look at, so you want to make sure, first of all, that all of your data has been fully compensated and you export your population of interest. So I'm not going to go through that today, but essentially what you want to do is you've got a file in FlowJo and you want to export your population of interest here. So you do your normal gating strategy. Let's say I just wanted to export my lymphocytes here, and you know that your file is good, and you're happy that it's all been properly compensated, and this is the only population that you're interested in doing at. Then you just right click and you can export, you can go through and export your populations here under export concatenate populations. So you want to export first all of your compensated data. So you go through here, you click on export. And that's what you want. You want to export all your compensated data. Everything that else is in here is the uncompensated. So for a bunch of these samples. So when you've done that, and you've now got all of your samples of interest, and these might be, you know, the total cells here, you pull them into the experiment, because what the next step we're wanting to do is to put them all together in one file, to concatenate them together, to do some high dimensional analysis. So these are now all of your exported populations of interest. So you want to come in here now and apply all the metadata that you want to these samples. So some of you may not be naming your parameters as you go in FlowJo, and there is a way to fix that here. If you go down to edit columns, you can add in all of your reagents here as columns. And experiment, I've labeled this differently, like this, for example, which I'm an OCD person, that really irritates me. But it will also mean that if there's a mismatch in the naming, when you concatenate the file, it will just drop the name. So you want to make sure that they're all together. Just a reminder to everyone, can you please mute? There's a little bit of background noise. So the way that you can do this as well is you can apply to all. If you name the top one, you can copy the value to group. So if you've got now 50 samples in here and you realize that, oh, actually my BV786 wasn't, it was actually XCR6, I misnamed it. If you highlight that and hit copy to group, it'll change all the way down. So this is a really good way to fix up your parameter naming if you didn't do it at the time in FlowJo, or you've got misnamed things all the way through. So do this first, make sure that everything's compatible, that you haven't accidentally put a file in here that's got a different parameter, because it won't be able to concatenate together. And then we can go and strip those back out of our view because it's messy. Okay, so I'm not interested in statistics at the moment. I'm just interested in these. So the next step that I like to do at this point is to put in any metadata that you're interested in into your samples that's going to help you to figure out changes in your clusters. So you just right click here and add a keyword. The default, it gives you sample ID. Don't use that because it automatically defaults to just kind of using sample ID to define each of these individual files that you're pulling in together. So something that you might want to put in is, say, a cohort sample ID. And in this instance, I've got 3 samples, and I would have a sheet to one side and say, right, 1 is going to be healthy, 2 is going to be ulcerative colitis, and 3 is going to be a Crohn's disease. And I would label each of these so that later on, I'm going to be able to actually gate on my different cohorts. So whatever parameter you can think of, convert it to a binary system and put it as a number. So some of these will be easy to do. So for example, age, you can just directly put in the age of the participant or the animal. The other ones that you could put in would be, say, sex, and you could have 1 as male, 2 as female. Don't use 0, start with an actual integer. So you could add in sex and have these as, 1, 2 female, 1 male. You could add anything else that you can think of. So for my cohorts, they have a disease score that's defined as the Harvey-Bradshaw index. Or you could actually just write disease score. Anything else that you can think of, you can put it in here as a keyword and just keep going until your heart's content. Anything that you might want to do later on in terms of statistical correlations, analyses, just put that in here now. So the next step that you're going to want to do once you've added in all of your metadata that you're interested in is now downsample these for concatenation. So the reason that we do this, you can see here the number of cells at the moment, it's quite different. And if you just double click on these column headings, they will reorder by what you've just clicked on. So I can see here now really quickly that this sample's got the smallest amount and it's at about 55,000. So in order now to make sure that I'm putting in equivalent amounts of each sample, I want to downsample to make sure that I've got 50,000 cells that I can stitch together. So the reason that you do this is if I was to concatenate all these files now, the overall population would be skewed towards populations that are evident in these samples rather than this one. So you want to stitch together everything evenly so that then the different clusters and the proportions of those clusters that you're seeing are representative of the entire data set and not skewed by individuals that had higher numbers of cells. So for this, we use a plugin called downsample, and all these plugins are under workspace plugins. Once you've installed them, you basically just restart FlowJo and it'll come up. So you run downsample. It'll tell you that before you do that, you have to save the workspace, because every time you run a plugin, it will create a separate folder where it writes everything to. So you tell it here how many events you want it to downsample to. So I'll say 50,000, and then you can just call it whatever you want. So this has now created a population here that is a representative sampling of the parent population, but with only 50,000 total events. And so we just want to now copy that to the group. And now we've got the 50,000 here everywhere. So to concatenate these files, you only want to now be concatenating your downsampled population. So if you click on this, what you can do is say select equivalent nodes. And what that'll do is select every group that's called down 50K in this group. So again, you can see how doing some of these quick tools is going to become really time saving when you've got 100 samples in here. So now we go into export concatenate populations, but instead of exporting this time, we're concatenating. So under concatenate, you're saying that you want to include all, because we've already done the downsampling. You tell it where you want it to save. So this will be good. So I might put it under, where have I got it? Actually on my desktop, FlowJo in here. And this time you're going to use the, I believe it's uncompensated, but let's just double check. No, still all compensated. There is a point where it becomes uncompensated, and I always get them confused, but this is how you just double check. So you want to have all the compensated parameters together. And now here's the important bit where you want to just, sorry, Song Yi, could you mute, please? It's coming through with quite a lot of background chatter.
Sorry.
Thank you. So now here's where you want to stitch everything together. So you're going to concat everything together and give it a prefix. So I might say concatenation 50K unstim, for example. And to make sure that now all your metadata comes in, this is the key point here. You want to add additional parameters. So you go into here and you go down and you find all these keywords that we've added in. So we wanted to add in sex, we wanted to add in disease score, cohort, age, any of the other keywords that you've defined, you need to add them here, right? And now when you concatenate together, and we're going to say load it into a new workspace. Now we've got all of those data here concatenated into one FCS file. And if I now put on the, keeps asking me to say, now if I put on the bottom axis, you can see I've got all these metadata here. So I can now split by cohort. And if I gated on population 1, that's all my healthies, population 2, that's all my Crohn's, population 3, that's all my ulcerative colitis. So at a quick look, you can even just quickly scroll through and have a look at the difference in some of your parameters. You could look at it, differences by sex, differences by whatever else you've put in here. All your metadata is now embedded into your FlowJo files. So this is also a really quick way to make sure that there aren't big outlying files that have some. See how I said not to use sample ID? So it automatically assigns that for each of the FCS files that you've concatenated into this sample. So you could do a quick scroll through. And by the way, if you're still doing this version to select the next thing that you want to look at, if you're on a Mac, if you hold down your command button and you just click, you're just going to scroll down through all of your parameters. You get down to the bottom. If you want to go back the other way, hold Shift and scroll, and you go back the other way. And when I learned this, it blew my mind and saved me so much time. So Shift and Command and click. And so you can see that they're all looking pretty good. So I now have my concatenated file that I want to play around in. So I will get rid of these examples, and I will now load up a concatenated file that I've prepared earlier, like a good cooking show. And we'll have a little bit of a play around with tSNE. Okay, so here's a concatenated file that I've played around with earlier. So you can see that it's already been gated on my population of interest, which happens to be CD3, CD4, double positive cells that are co-expressing these activation markers that are an antigen-specific cell population. So this is another quick way just to go through your gating strategy and make sure you haven't accidentally concatenated a double negative file or something. So they're all there. There are cells of interest. So the first thing that you're going to want to do now is run a tSNE, and that's native to FlowJo. You just run the program here. It'll, again, you have to save it first. So it has somewhere to write the data to, because there's a lot of things that happen in R in the background. You don't need to have R open. Okay, so then it pops up here and it asks you what parameters you want to use. Now I've deselected the parameters that I know are my CD3, CD4, and my other two. So I've already gated on the cells to all be positive for those markers. So I'm not really interested in how they cluster or not. So I've deselected them. There are ways that you can play around with these iterations. I just leave it for now as the default settings, and then you hit run. So the more cells that you have in your concatenated file, the longer tSNE will take. I've got just under 4,000, so this is going to run in a couple of seconds. You have a couple of million, you know, depending on the speed of your computer, you might need to walk away for 15 minutes or so. All right. And so now this is what the concatenated file looks like on a tSNE plot. So just a reminder that tSNE is dimensionality reduction. It is not clustering analysis. So next we want to do some clustering analysis to figure out what unique biological data clusters might be in our data set. And you want to be doing all this on your parent concatenated sample that includes all of your healthy and your disease and all your other cohorts, because it's taking all that into consideration when it does the clustering, and then later you can manually separate and see if clusters are different between a control and a population or a treated sample or something. So you come back to your concatenated sample and you'll notice that your plugins are grayed out or they won't work, and you have to have an FCS file highlighted to run a plugin. So I highlight back my sample and I'm going to run Phenograph today. FlowSOM's another equivalent. And again, here you tell it what populations of interest are. So I'm just going to take the ones that I'm interested in here. And again, you can change the K value, which is about how many clusters it's going to find, or the rest of it. And you can go and read up about that. But again, I just leave it at the default. And I just hit OK. So it's now running Phenograph in the background, and that'll take a few minutes. So the other thing that's useful to know is when you have a plot that's open like this in FlowJo and you want to put it straight into the layout editor, on a Mac, if you just do Apple L, it'll just dump it straight into the layout editor for you, which is really useful. If you wanted to, yeah, if this changes and you wanted to put, for some reason, that one in, L, and then you've got your second one in there. So I find that this is really useful initially when you just wanting to have a few different views and just dump them straight into FlowJo. And you can see here that I've got my tSNE axes down the bottom here. So once you've run tSNE, it links it to this sample, and you've got these as axes now on the bottom. The color axis can be interesting to have a bit of a look around as well. It basically colors by density of expression of these markers. I think it used to be a little bit easier to read in some earlier versions of FlowJo, but regardless, you can play around with it a little bit. And so the other thing that we're going to want to do eventually is actually gate on our sample populations here. So my healthy and my Crohn's and my colitis. So you can just put gates around as you would normally do. This, is that not working? Probably because it's in the middle of running something else. So the other way, so now Phenograph has given me these 8 populations that it's found in the data set. Now that you've got your tSNE map that you've dumped into your layout editor, you can just directly drag and drop those populations on top here, and you can see where it's defined the clusters. Okay, the count may not be useful, so you can go into here and say, right, instead of the count, I want something else. I want the, I find the proportion, so the frequency of parent is probably more useful than the count or the name here. The sample name's not really useful because that's the same for everything. And then you've got your frequency here of parent. If you don't want to have that ungated population there, you can just right click and remove that layer. Okay, so then the next one to have a quick look at is ClusterExplorer. So again, under plugins, ClusterExplorer, you again tell it which ones you're interested in. It needs to have a tSNE map and it needs to have clustered parameters. So you have to have run a Phenograph or a FlowSOM clustering beforehand, and then hit OK. And this is what it gives you to play around with. So in the 8 clusters that it's got, of all the markers here that I'm interested in, here's what they look like. Here's why they're different. And you get this great heat map down here. And so if I was interested in the populations that had the highest TIGIT, for example, I could double click on here and just look at them and go, right, it's population 1, then 7. If you just want to look at 1, you can click on that and go, okay, population 1 that's got the highest TIGIT is, you know, lower for PD1 and lower for 226, but higher for CCR6. You go, what's that population there that's really low for CCR6? And it might be this one here. So then you can start playing around and interrogating your clusters and figuring out actually what their phenotype is. So, you know, this one here that I was interested in, it's quite low for CCR6, but it's quite high for integrin beta 7. And if I'm interested in integrin beta 7, then it's this one here that's also high for CCR6 and high for integrin beta 7, which is the population that I ended up being interested in. So once you've done that, and I might just open one that I've done up earlier here because the gating's being a little bit difficult. Right. So you can see here, I've put my gates on my 3 populations, healthy, Crohn's, colitis. I've run in this instance, the FlowSOM. I've got my populations of interest, and I ended up defining that, that CCR6 integrin beta 7 as potentially a population of interest. And I just kind of set the gate on my concatenated population where I thought that cutoff would be. Then what I've done is I've gated on my 3 populations of interest. So my healthy, which was defined sample number 1, Crohn's that I defined with a keyword as sample number 2, and colitis that I defined as number 3. So I just gated on these 3 populations and named them. Then what you want to do is actually export these populations now as individual FCS files to do side-by-side analysis. So the same as what we did before, you just export the populations and then you go through the populations here. I think this is where we switch to all uncompensated. Yeah, so you can see here, if I select compensated, all I'm getting is forward and side scatter. At this point, it's kind of the compensation's embedded, but I haven't added extra compensation. So you can see here, FlowJo comp, you want it all uncompensated. Okay, yes, you can name it whatever, export, and then you can ask it when you do that to put it back into the same population, into the same workspace, which is what I've done here. And you can see I've called it post-tSNE concatenated healthy, Crohn's, and colitis. So now these three gated populations are three individual FCS files. I created now a new group up here called post-tSNE, and I've just dragged and dropped those exported populations into here. Now I've taken across my FlowSOM populations that came from my total concatenated analysis here, and I just dragged and dropped these onto my exported populations so I could do a side-by-side analysis. So now what I can do is open up, and every time that you've done this, you've now kept your tSNE things on the label, the tSNE axes. So by doing it this way, by doing the tSNE analysis first or your clustering analysis first, and then exporting, all of that tSNE analysis gets linked to your individual samples. And now you can actually start having a look at what's different between your populations. So when you come into your layout editor here, I've dragged and dropped on the overlay here my clustered populations. I've looked at the frequency of parent, and now I'm looking at healthy versus Crohn's versus colitis. And I can look down here and look at the frequency and looking at what's changing and visually looking at what's changing and start picking out populations that might be of interest. For example, population 6 here has gone from 5% in healthies, and it's basically doubled in my 2 disease populations. And you really want to be looking at least a doubling at this stage when everything's concatenated together before you start going back to all of your original raw files, applying a new gating strategy and looking statistically. Because by the time that you go back to each sample one by one and apply a new gating strategy and try and understand if there's a statistical difference, you really want to be starting with at least a doubling difference here, a fold change of 2 before you're going to find anything. So this is how you get those nice overlays that you see in some of the plots. If you've defined your own manual gates, you can also just drag and drop them directly onto your tSNE map, and it will create a new layer and you can label it. The way I've done this here too is it's another way of interrogating your data. If you're not using ClusterExplorer, you can just right click and put these multigraphs here on the side and create histograms and adjust the ones that you're interested in doing and the populations that you're interested in looking at. But now that we've got ClusterExplorer, I find that's a much easier tool to figure out what your populations are looking like. So in a half hour, that's really all we've got time for today. So I hope that's given you guys a bit of an overview as to how you can start getting some more data. And the beautiful thing about this at the end now is that because you have all that metadata linked to all of your samples at the very beginning, if you now go back to the beginning and you want to look at exporting a table editor with like all of your MFIs or all of your population percentages, at the same time that you do that and you say create a table, what you can do here is edit the table to also show your keywords. So when you go here to edit column, you can add your keywords into this that you had for your samples. So for HBI, for example, so that's my Harvey-Bradshaw index, or my sample number, which is my details of my cohort. I can add these into my table export at the very beginning. And then when you create a table, all of those metadata are linked to all your MFIs and everything. And when you copy and paste into Excel, you can just filter by that and do really quick correlation analyses of whether or not the MFI of protein X correlates with disease score, for example. So happy to take any quick questions now. Otherwise, feel free to come and drop by level 7, the Villadangos lab, or send me an email. I'm happy to do one-on-one walkthroughs with this stuff.
I got one simple question. Once you get the, can you hear me?
Mm-hmm.
Yeah. So once you get the different populations that come out of phonograph or these ones, you export them because you want to then be able to do things like batch analysis and get frequencies and comparing different, apply different gating, apply essentially the same gating strategy, but getting the statistics out of each sample individually, no? Because you could also set up gates directly there, no?
Yeah, it's easier to work with them as an FCS file because that way
you'll be an independent sample in the,
it's treating them in, and then you can do like your if you create the gates here, you can just copy and paste them everywhere. It's just a lot easier and cleaner because then your frequencies of parent are of that total population from those cells. So you want to do all of your overarching analysis, so your discovery pipeline, you want to do on everything together, so you're not biasing towards clusters that might be present in control versus comparator. But once you've done all that, then to do your exploration, it's just easier to export them as individual FCS files, put them into a group, and then you can do group analyses and compare percentages a lot easier.
How long does it take you to do, imagine that you're going full on in the number of cells that you want to analyze. How long will it take you normally to do the whole procedure with, I don't know, imagine 50,000 cells per sample or something?
So the longest thing is the very beginning step. So in each experiment, opening up, making sure it's all compensated, doing the gating, and then right-clicking and exporting population of interest to be concatenated into a separate folder. That's the pain in the ass step. But you put all the time and the effort into doing that, and then it's just a matter of pulling all those files in. The other thing I didn't mention is that when you do those first samples, for those that might not have seen here in my preferences, I have my naming set up to be a keyword combination here of the tube name plus $SRC. So that keyword, that $SRC in Diva refers to the specimen name that you've allocated. So if you've said specimen name is, say, mouse 1, and tube name is like unstim and stim. So now my name here is my specimen name plus my tube name. So I've already defaulted that here. The other way you can do that is go in and just find $SRC wherever it lives down here and put it in as a keyword. Where is it? And you can Google these up on different cytometers. It'll be different names. Oh, I forget where it is, but anyway, you can. I think you can find it, and there it is. $SRC. You put it in here, and you can also change this. So you can see if you accidentally forgot to name it or you misnamed it when you exported your data. You can fix it here and say, oh, actually, this was patient 3, not patient 2. So you can do all that there when you're concatenating at the very beginning. And
once you export it, that the keyword will be corrected.
So once you concatenate these files, all these data will be linked into it, and then coming back here, you can fiddle around with it. Andrea, you've got a question? Hand up.
Yeah, thank you. That was amazing, Laura. I just got a curiosity. When you've shown outside the application of the phenocluster or the phenograph, the clustering analysis, you've shown like something like a heat map. Based on what does the system? So based on what does the system recognize what's a high expression or low expression I'm used to do like negative versus positive population. How does the software recognize an upregulation versus downregulation? Does it make sense?
So it's just based on at the moment, like what's in your sample. So for example, in my concatenated sample, you can look at the spread of, oh, come on mouse, Jesus Christ. You can look at the spread of this expression, for example. Here's low, but there's the highest. So you can kind of see based on the data that you're giving it, that's the range, right? So if you've already got in here, let's say you added your keyword at the very beginning, and number 1 is your like baseline or your unstim, and number 2 is your stim. And then you could quickly have a look here and look at, okay, yep, this marker should have gone up, and here it is up here. So then basically what the system will be looking at is just it's a spread of those data. So the heat map analysis is looking at what is the expression of these different markers in what the clustering algorithm has determined to be a unique cluster. So depending on how many parameters you're looking at will depend on how many clusters you get. So if you're looking at only 5 parameters, you might only get 5 or 6 clusters that make biological sense. If you've got 20 parameters, you might end up with 40 clusters that biologically make sense. So when you run that plugin under the ClusterExplorer, all it's doing is looking at these 8 populations that have clustered based on what you've inputted. So I've told it these 8 parameters on what I want you to use to define clusters. And then the ClusterExplorer is basically giving you an idea as to what's unique between those clusters. Oh, this cluster that looks really interesting is actually got the highest expression of this marker and the lowest expression of this marker, but that's only based off what's in your sample, what you've asked it to look at. Does that make sense?
Yeah, thank you. Perfect. Thank you.
Cool. Any other questions, or was there anything else that didn't really make sense?
I don't know if there is still some time, but I would like to ask you if it's possible to repeat the step from the analysis with the phenograph to the moment you have the different cohorts in 3 different plots. I didn't really catch that.
Okay, so that one here is when you've got your concatenated population here, you want to draw a group, basically. You want a gate on your population of interest. So this is, that's going to be my healthy, because again, I've previously defined this keyword. These are all my healthy samples. I defined that these were all my, Crohn's samples, and then these were all my colitis samples, for example. But, yours could be different. You could have 5 groups, you could have 2 groups, but you've got a map somewhere that you know what these relate to. So you've now got these 3 groups here, and you can see they've come up here. So the next step that you want to do is to export them as individual FCS files. Sorry, my mouse is being a little bit silly here. All right, so you want to export those populations here. You want to go with, this was where it was all uncompensated parameters, and you can export them. I just kind of call them export post.
Select export. I think you're in concatenate, no?
Yeah, sorry, export, thank you. Yep. Yeah, so export post tSNE in the export tab, and then it will say here it's going to generate you 3 new data files, and then you just hit export, and I say put them back into my existing workspace and close, and here they are. So now these 3 gates are now existing as 3 separate files. So when I do the sample number here, yeah, you can see that's number 3. Right? So now you've got those as 3 separate files. What I do here is create a group that I just call post tSNE, which is here. And I just grab these 3 files and put them in here to start doing collective analyses. So you can see that they've now got the tSNE axes linked to them. They've got the phenograph analysis linked to them. They've got all the keywords that we put in the beginning linked to them. So if you wanted to have a quick look in the data as to whether or not sex made a big effect on a marker of interest, you could just put that here on your x-axis and have a quick look as you scroll through the axis here. And yeah, so I didn't put any values in for HBI in this instance, but you know, there's my sample IDs, and you know, do I have an outlying sample for any of these markers? And you can just, you know, quickly scroll through kind of thing. So that's the way to do it. And then once you've got the these over here, then all these populations that we predefined, you can now just drag and drop them, right? And it treats them like grouped analyses. And then you open this guy up, and I'm going to do the tSNE gates on here, not time, tSNE. So sometimes it does this as well, and you just have to go and customize the axis and just, what was it meant to be? I think I go back to logarithmic for this and change it back here. I forget each time, but it does silly things, or it's
not linear. Go for linear.
Linear, okay.
Yeah, minus something to plus something typically.
Yeah, so FlowJo sometimes just stuffs up the axis here. There we go. Thank you. Okay, there we go. Okay, so now I've got that there. I'll just do my Command L, throw it into the workspace, and then I can drag my populations on here. Get rid of the stuff here that I don't want, add in my frequency of parent, get rid of that one, hit OK, make it look the way that you want it to look, and then just hit batch, and you're going to do a batch for your post tSNE, and you know that now you've got, that's your healthy, that's your Crohn's, and that's your colitis. And so you can now have a quick look down everywhere and look at your look at your changes in your percentages and figure out which one of these clustered populations may be of interest. You know, there's one here that's missing in one of the populations, for example, and then that's where you go and you start playing around with ClusterExplorer to look at the phenotype of these clusters and have a think about, right, well, which ones are changing drastically by at least two that look like they're really different between my cohorts, but then biologically, does that make sense? Is that even an interesting population? Yeah, does that make sense?
Yeah, perfect, complete, very clear. Thank you so much, Laura.
No worries.
Just another question, Laura. You can treat this exporting tool and importing as a file as a way to essentially export all the samples that are within a given group. So essentially, you treat the group as an independent file to see global changes between groups before you go into specifics. No, to see actually if there is you don't have an outlier in the group or something like that. Yeah,
exactly.
Yeah,
so yeah, and then once I've done this and I've figured out that, you know, for me, my CCR6 integrin beta 7 gate was the one that I was most interested in. At that point, I want to go back to, so this was my gate that was the one that I found of interest. That's then when I go back to my downsampled at my original populations, right? And I can actually just take that gate that I defined. The fun thing is you can kind of use these FlowSOM populations to do almost like a purity check. If you think that population 6 is of interest, you can start playing around with what is my minimum gating strategy to capture like 80–90% of that population of interest. This was the gate I had of interest. I can now drag and drop that back over here. Yeah, okay. But I've defined what that gate's going to be. I can come into my original samples and I can apply that gate essentially. And then I can export it. So I can come back to my original samples here, because now I'm doing an actual gating on all the cells, and define a new gating strategy, and I can come back here and export that with the table editor with adding in all my other keywords. So in the end, I end up with this Excel table export that's going to have sample name, it's going to have cohort ID, age, sex, disease score, everything else that I'm interested in as a clinical parameter, plus MFI, population percentage, blah, 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 blah. And just boom, in one big spreadsheet. And then you can just plug and play into Prism or your stats thing of interest.
Yes. So yeah, I can ask a question. Anyone have any other question to ask? Because I have another question for Laura. How many times, something that I was looking at the way you do the discovery and then go back and define the gating strategy, as you mentioned. I have thought in the past to use high dimensionality reduction algorithm and clustering to define gating strategies that I'm not aware of. So if I have a sample that have been stained with 12 colors and I don't see actually a clear two-dimensional hierarchy that gives me a nice resolution of population that I know and clearly distinct, I thought, well, maybe I should do the analysis of clusters and then go back and find the gatings. Yeah,
that's exactly what I do. Yeah,
but something that we need to be, I mean, something that I also have been aware of since doing the analysis of autofluorescent and stain samples is sometimes these methods are more powerful depending on the method you choose. The method could be more powerful than the traditional two-dimensional approach. Yeah. So you can end up with operations that are slightly overlapping, but they are still distinct based on the analysis of the high dimensional reduction and the clustering analysis. Yeah.
So for example, I was, my pipeline previous to this had always been a hierarchical gating strategy where you're kind of looking for what you're already looking for. Yeah, I'm going to gate on my CD3, CD4, CD3, CD8, and then I'm going to look at this and then this and then this. And so you're going down that pipeline and you're doing that in all of your individual FlowJo samples and you're exporting data somewhere and then you're looking for differences. And that's great. And you might still want to validate with a manual gating approach, but in order of trying to find changes in populations that you might not have thought to look at. Like, for example, I wasn't thinking about looking at beta-7 in combination with another chemokine receptor, or I might not have been interested in looking at that on the CD8s. But by coming in here and just saying, like, I'm interested in changes in T cells, and just concatenate all my T cells together, total CD3 positive cells, downsampled to say 50,000 cells per population, come in here and run this, and looking at the clusters that have come out, there could be a really unique population that comes out that I would never have thought to gate on. And by doing that, and like I said, on this total population here, if population 4 is of interest, you can kind of, using your clustering analysis first to have an idea of what the expression is of this marker, then you could define a minimum gating strategy and you think, right, so if I get to my total CD3 positive cells, then what is like 1 or 2 gates that I could do to define this population by say 90%? And you could say, oh, I'll try this gate, and then if I take those, maybe I then purify it by something else. And by doing this on the clustered group, you can kind of use it, like I said, kind of like a purity check, in that if I set the gate here, I'm getting 70–80% of that clustered population. And then you can kind of think, all right, well, that seems to be a biologically interesting population. You define a new gating strategy for it, but then you still need to go back to all of your individual sample files and apply this as a new gating strategy to get your percentages, to then put into a plot, to then do the statistical analysis to look at, including all the variance across my entire cohort, is this now a statistically significant difference in the expression? But this is a really powerful discovery tool that can help break you out of that mold of, I always do the same gating strategy because I think I know what I'm interested in. And instead you're asking the data to show you what is changing without you putting your predefined gates on it.
Perfect.
Cool. Well, it looks like we've kept most people for nearly an hour now, Alexis. We might have to change the time frame.
Yes.
So I hope, so please give us feedback, guys, if this is useful, if this is the kind of thing that you want to see. I had it half an hour initially, so I did kind of rush through it, but I'm happy to do a part 2 version where we can go through some of the stuff a little bit slower, and particularly the next phase, which is kind of validating some of these findings and going through that second part of the workflow.
I think the strategy of the cooking show works perfectly, having some things already made. So you don't have to go through the whole calculation time. That was awesome. But then I think probably we can think of doing another one slowly and maybe based on your actual results. I mean, exploring more into these results that you're saying, on the whole story of it, how you went from initial assumption to the final discovery of a population that you were not intending to.
Yeah.
So
exactly.
All these seminars, I mean, with the approval of the lecturer, I will be posting the seminars in the YouTube channel that we have, and the links to the channel, to the lectures, will be in Capendo. So in Capendo, in the first page of Capendo, you have resources, and normally I will put in there links and documents concerning information for Aurora, spectral mixing, instrument, advanced analysis. So the links will be there. And I'll let you know once the video is ready. Thank you, Laura, so much.
Yeah, I was just going to show, this is kind of the end result here that we got to, and it's published. Nice. So, you know, found this unique cluster here through the tSNE analysis, went and defined a unique gating strategy, and then went and did the statistical comparisons here and found some really interesting correlations on these antigen specific cohorts. So that's how this workflow ended up in a publication.
Nice.
Yeah. Okay, so yeah, and likewise, I'm just l.cook@unimel.edu.au. So feel free to, like I said, come and knock on the door up on level 7, or shoot me an email. Happy to walk you through this. If you run into technical difficulties with your plugins, just reach out to FlowJo tech support. They're pretty responsive.
Perfect. Goodbye, everyone, then.
Thanks, everyone.
Cheers. Thanks, Laura.